Hi, Professor. So I'm going to do the think aloud. So, cellular structures. Cytoplasm is a matrix found between the nuclear and the plasma membranes in which chemical reactions can occur. So right here, we can see that cytoplasm is in the nuclear and between the nuclear and the plasma membrane. So we know that it's this area in the cell. Um, and so Um, the structures of the cytoplasm is water, ions, proteins, lipids, carbohydrates. So here, water makes up 70 to 85% of cell mass, and we have different types of ions. We have magnesium, chloride, very common, calcium, phosphate, proteins, structures of examples that are we can think of that are part of the cytoplasm or that make up the cytoplasm is the microtubules, the microfilaments, and they're arranged in a complex lattice work that functions as the cytoskeleton. So actually, I'm sorry. So actually, that's the cytoskeleton. So the cytoskeleton is a part of the cytoplasm. And it provides the structural organization, and so it's basically what holds our organelles and our cell in place. Enzymes are proteins that play a role in the metabolic functions of the cell. So from what I know about enzymes, they catalyze, they help to speed up chemical reactions. So they lower the activation energy or the activation that is required. So if without enzymes, it requires this amount of activation energy to um, undergo metabolic reactions. Enzymes decrease that because of the rate in which the chemical reactions can occur. So that's something to know about enzymes, and that's why we need them, right? Because we want to be able to do these chemical reactions really fast in our body. And we have lipids, we have the triglycerides, phospholipids, steroids, and prostaglandins. That's the first time I'm um, learning about that specific type of lipid. Okay, so we're gonna go into for carbohydrates. I didn't have much under carbohydrates. Um, And so free ribosomes, they're composed of protein or ribosomal RNA that synthesize protein that is used inside the wall. So ribosomes are composed of okay, this is very important. They're composed of two subunits, um, 60S subunit and 40S subunit. We have the endoplasm reticulum is a system of membrane-bound canals or tubes. So we have the rough ER and then we have the smooth ER. Um, the rough ER has ribomes attached to it. So I kind of think of like, okay, it's rough. So that means like it has things that make up the roughness, and that's the ribosome. That's my way of thinking of it. Examples where we see rough ER is in plasma cells that help to synthesize antibodies that are released into our bloodstream. And rough ER do not have any ribosomes attached, and they function in synthesizing steroids and lipids and storing calcium. Um, so that's a very distinct function of a smooth ER versus a rough ER synthesizes proteins to be exported out of the cell. Okay, the Golgi apparatus. One way of me memorizing Golgi is thinking about Amazon and how Amazon is like the package center, and like so basically Golgi is like where like the proteins are sent to be packaged and be sent out. So like think about like if you received your Amazon package without it being in a package, you would just wonder like why do they not have a package around it? And that's the same thing with the protein, and that's the function of the Golgi apparatus. Um, So it's also complex in that it adds carbohydrates to the proteins, forming glycoproteins, and to package the protein. So that's a very important aspect of how they package the proteins. Okay, the mitochondrion. So basically, one thing I've learned is that it's the power center of the cell, it or powerhouse of the cell. Um it basically is our the energy currency for the cell. This is our cells need energy, and so energy is made from energy in the form of ATP, and so that is where it's made. And there's a process that it goes through: glycolysis, the Krebs cycle, um, the electron center, to produce for one glucose molecule. It can produce about 36 ATP. That's a lot of energy for one glucose molecule. Okay, so lysosome. So basically, I've learned in the past that lysosome is basically, it kind of has digestive enzymes that break things down, and basically is like the cleaner of the cell, the cleaning center of the cell. So if you think about your mom and pop shop, you take your dirty clothes to dry cleaners and they clean it for you, and so basically that's the image that I have of lysosome. So they're a membrane-bound sac that contains 60 different types of enzymes. That's very important to know. That's a lot of different types of variations of enzymes. And it's needed for proper turnover of glycogen and certain lipids, and lack of these enzymes can result in abnormal accumulation of these molecules in the cell. So the lysosome actually contributes to a very, very needed part of our body's mechanism. That's another important thing to note, is that the POP disease results from the accumulation of glycogen in the heart and the skeletal cells due to the lack of lysosomal enzyme. And I'm assuming that that comes from things being built up because they don't have these digestive enzymes that are able to break these down and basically like recycle and get rid of and dump the waste that's happening in our cells. I'm assuming that's something to think about, right? So we have the microtubules. They're these thin hollow tubes. They're 25 in diameter, 25, I'm assuming it's nanometer in diameter. And so basically they form centrioles, they form cilia and flagellum, transporting materials within the cytoplasm. Centriole consists of a rod-like centrioles positioned at the right angles to each other. And centrioles form spindle fibers, thus aiding in the movement of the chromosomes during cell division. So this is very important aspect of cell division. Sorry, give me a second. My laptop is about to die. So that's important to note, that that centrioles they help in the aid of cell division, and so that's an important part of having this as part of our chromosomes. Okay, so we have the cilia. They're tiny hair-like structures that basically what I can think of cilia is that they're these tiny little structures, hair-like structures, and they kind of help to move things in the cell. So they move to function to move substances across the cell surface. Yep, like it's saying. And cilia are composed of microtubules arranged in 9 groups of doublets with 2 microtubules in the center. So that's how it's arranged. Flagellum is a single long structure that protrudes from the cell. So if you think of a cell like this, the flagellum is like something that like looks like a little like not straight, but like wiggly. And basically it's to like help propel the cell, right? And in humans, we see this in sperm cells, and that's kind of like in human cells where we see it, versus in it's very more common in prokaryotic bacteria cells where we see this structure very much common, versus in our cells we only see it in the sperm cell. Microvilli are brush border, so they're finger-like extensions of the plasma membrane, which increase the surface area of the plasma membrane and function to increase the absorption capacity of the cell. Um, so I could just think of like a villi. That's like usually villi are like these nice big houses. And so just think about the surface area that that house covers. I could just think about microvilli to make that association, and kind of think about that they increase the absorption and capacity of the cells. Think about these like huge villi buildings covering the surface area of the cell, and that just to like jog my memory. Vacuoles are um, membrane sac bound membrane bound sacs which store substances within the cytoplasm. Um, and that's yeah, um, that's that's pretty much that's what it is. Secretory granules or vesicles are membrane bound sacs containing substances which will be exported out of the cell. So this is important because it's important to note that substances that like will be exported out of the cell, so movement out of the cell. So we can, if we were to categorize this, we would put this in the category of things that aid in moving things out of the cell. Peroxisomes are they're spherical membrane enclosed organelles which contain several enzymes that detoxify harmful molecules that break down hydrogen peroxide. Um, it's also the enzyme catalase within the peroxisome that breaks down the hydrogen peroxide to water the oxygen gas, thus preventing the accumulation of hydrogen peroxide. The fission, when hydrogen peroxide is poured on a wound, is the release of the oxygen gas that occurs when catalase breaks down the hydrogen peroxide. And this is important because these enzymes help to detoxify harmful molecules in our body, um, like hydrogen peroxide. So that's very important to note the function of this. And that concludes the Think Aloud. I'm trying to figure out how to save the recording.